this for a minute, this makes some sense. Every time you eat a meal, you are eating antigens that are not yours. Every plant, every animal has shapes on the cells. Why don't you go attack a Whopper when you eat one? Because my helper T cells learn to ignore my meals. Otherwise, I'd be attacking everything I ate and eventually die. So your helper T cells choose to ignore the stuff you eat. Or, if you're pregnant for 9 months, why don't you kill your baby every time? Baby's not yours, right? Its antigens are not yours. Why don't you kill your own child? Because your helper T cells choose to ignore it. Right? So, your helper T cells have a choice. They can choose to attack, or they can choose to ignore. Right? That's based on the helper T cells.
So is this where food sensitivity after a fast can come from?
Yes. This is also where they talk about tolerance training. So you can teach your helper T cells to ignore things over time, like your children. Yeah, in a bigger sense. Yeah. Worthless trivia, I don't know if I mentioned it before, but I have honeybees, and the rule in beekeeping is you must be stung every year, multiple times. Because every time you get stung, you hope that your helper T cells keep ignoring this thing. The more times you get stung, the less of reaction I get. It still hurts, don't get me wrong. It hurts every time a bee stings you. But you swell up less and less because I'm developing tolerance to the honeybees.
Why do some people swell up more and more every time?
That means you're not tolerant anymore. You are this now. So once you start to swell more and more, you stop being a beekeeper. Because you're going to die the next time the honeybee stings you. All right? So that's tolerance. Yes.
And fish allergies too.
That's right. Very good. That's the next line here. Is there's a third choice your helper T cells can do, which is called the hypersensitive response. Hyper meaning too much. You're too sensitive. You know this as allergy. So an allergy is a decision by the helper T cells to overreact. You went nuclear over peanut butter, right? So a normal response is competence, ignoring the thing is tolerance, and going wacko for peanut butter is allergy or hyper response. But the helper T cell is what's choosing, quote, quote, that idea. So what we're going to do is show you a brief picture here of an allergy in motion. Here we go. At the top, what is that thing in the squiggly lines with the dots? The answer is always on the screen. Those are bumps on a cell, those are antigens. Okay, so that could be peanut butter, that could be fleas on your dog, that could be whatever. Okay? Notice this arrow. We went from this thing to plasma cell. Remind me what plasma cells are, what they do.
They can make antibodies.
Make antibodies. How do they know to do that? Who tells them to make antibodies? The helper T cells. Notice what antibodies this plasma cell was ordered to make. Which ones? IgEs. Remind me what IgEs are for? This choice. So the helper T cell said, thou shalt do allergy. Instead of normal and ignore, I'm choosing to tell your body to go ballistic and make IgEs. The IgEs then visit this man, Mr. Basophil. Remind me from your lab exam, coming up, or half-ad, what do basophils do?
Histamines.
They release histamine. What does histamine make you do?
Swell and itch.
Do all that. Swell and itch and go. So you're allergic because you're swelling up and leaking fluid. Because the histamines were released from the basophil because the IgEs told them to. Right? So what you're seeing in an allergy is you're overreacting to something. The helper T cells are ordering IgEs, and the IgEs are telling your body to blow it up. And that's your allergic response.
So do they only ask to make IgEs? Or what's the relationship with that? And other times they're like, no, don't make an IgE, make this other antibody.
That's the helper T cells call.
But are there times when it does make an IgE and it's not?
No, IgEs are always histaminic. So you'll always inflame with IgEs. You may not call it an allergy yet, but you're always going to have this.
But there are some times where it might not be like a pathological condition.
Almost always it becomes one.
Okay. Okay.
I have a question. So, this is related to allergies, but I had a reaction to almonds, and then I stopped eating them, and then I started, now I can't have them because it starts closing down my
Yep, that's
system.
allergy. Yep.
So, because I didn't keep like taking almonds, that's like I didn't remember it, like my.
That's called tolerance training.
Okay.
So what they found out at Harvard was if you think you're allergic, like to peanut butter, that immunologists, people, white coats and big degrees, can teach your helper T cells to become tolerant. So what they do is they give you a little bit of peanut butter over a period of months and years, little by little, to train your army not to attack it. It's like the honeybee sting. Don't do this yourself, because if you do it yourself, you'll be dead. Because you'll do that to the point of being anaphylaxis. But about half of people who claim they're allergic can be tolerating the peanut butter, according to Harvard, if they get taught by immunologists. Right? So if you give up almonds or peanut butter, you're not going to have any tolerance, although you'll still be alive, which is good. Yes?
So my daughter had, she had bacterial pneumonia a bunch of times when she was young, and so they gave her penicillin, she had it a bunch of times, and then all of a sudden she broke out in hives the last time she had it, and they said she was allergic to it now, so why would she be tolerant to it, taking it so many times?
Blame her helper T cells. They decide they don't like penicillin. Unfortunately, I have no magical answer.
Yeah,
Your helper T cells
I
call.
just didn't know why she wouldn't be tolerant from taking it.
Ask your army.
Are there certain groups of antigens like protein shapes that will, set off the helper T? Like, I don't know, like I'm allergic to tomatoes and strawberries.
Yes.
Do they have like
There's been no connection made between antigens, because in theory you can be like peanut butter doesn't share anything with strawberry per se, other than it's plant. Dander, there doesn't seem to be a rhyme or reason of why certain things, other than the commonality. The more likely you encounter it, the more likely you'll show the allergic response, just because you're going to get symptoms more often. But there doesn't seem to be any pattern of why you're allergic to
I know
penicillin
there's a
versus
lot of
peanut
people that have, like, there's 3 foods that we all have the same allergy to.
Milk.
Sure.
That's just genetics.
If we want to know about how to make people more tolerant to allergens, where would we go?
Talk to an immunologist.
Immunology? Okay.
Yes. Because this is not something like the bee sting thing. You don't go out in the bee yard the first day of the year by yourself. Because that could be the one day when I'm no longer tolerant, and then I end up on the ground, dying because I start to break out. So you don't want to play with immunology just willy-nilly. You can try it at home, don't do that. But trained people can do this. So when they give you an allergy test, they're injecting you with the allergen and trying to see if you start to do this under your skin. And depending on where you inflame, tells them where the histamine was, which tells you where the IgEs were, which tells you all that. Okay? So allergy and tolerance are basically an immune response. Make sense? Yeah.
I read that you're not supposed to give babies like under 1 peanut butter or honey. Does that have anything
That's
to do
actually been
with,
disproven now.
Oh, really?
They actually think withholding it actually causes more of an allergic response than
And
giving
I
it to them.
think
Same
that's
story.
botulism.
Well, honey you can't give them to, but not
Yeah,
give.
don't.
So, if that were true, think about your grandparents should all be dead, right?
Yeah.
No one told them to ever withhold anything, and they're all quite fine. So it seems to be more reaction than science.
Oh, I have another question.
Yeah.
So with allergies,
Uh-huh.
I heard that if you take honey in your region, that it will help your allergies. Is that true?
If you're a child, yes.
But not when
Seems
you're
to.
adult.
Once you have the allergy, honey doesn't necessarily help much. You will develop tolerance, but that is true. Not that I'm trying to sell you honey. Right? Well, if you want to buy some, give me a call.
If you have some.
I can.
Jordan, bring it to school.
Bring it on the first day of class.
Bring it next week.
That's considered bribery, isn't it? If I bring sweet things for you.
Oh,
Oh yeah, that's right. Just bribe me. The general idea, like for example, poison oak. If you get honey from bees that have visited poison oak, your whole life, you tend to have reduced sensitivity to poison oak. But it's a real general anecdotal thing. You don't want to do that as an immunological treatment. But it tends to be, if you have foods from the local area, you tend to become tolerant if that's what you eat your whole life, in general. Yes?
What makes the T cells decide which response to
If we knew that, we could solve all the world's ills.
do? So it's just by chance?
Just by their chance, genetics, exposure. No one has the answer. I don't have the answer. Right? So there's different hypotheses, but none of them are shown to be true. So get out your packet of doom. We're going to learn a little bit about allergy together in the class. Find this page.
packet of gum.
33 on this one, a different page in mine. So it says, what does it say? That big word there. The goal of this case study is to do two things. One is we're going to learn allergies a little more. And PC says, thou shalt be able to read and interpret a scientific article in your field. So you're going to have to whittle down this gobbledygook that you might read someday as a future nurse, or not. Okay, but let's do some of these questions here as a class that are important, like A1. Remind me again what antigen is, because I'm going to ask you that on your final. What is an antigen? Protein where? Surface of a cell. That's the most important. If you get anything out of the last 4 weeks, it's on the surface of the cell. Okay? That's the fundamental concept of biology. So what the heck is allergen then, Marty?
An item you put into your system.
Something we're
Find out.
sensitive to?
This one actually used both interchangeably, and they are interchangeably. An antigen and an allergen are exactly the same biology. They're a shape on the surface of a cell. There's no biological difference between them. The only difference is what it does to you. So if we go to our little chart here, we tend to say antigens are what you're competent to, and allergens are what you're allergic to. But they're the same thing. So when you eat peanut butter, you're eating peanut antigens. If you become allergic to peanut butter, you're magically eating peanut butter allergens. They're exactly the same thing. There's no difference other than the response your body does to them. Make sense? So all it is a difference in degree, not the thing. So that right there are both antigens and allergens depending on how you deal with it. Make sense? So you can say antigen on a test, and that's perfectly legit. Basically, allergy generating response. Make sense?
So, I'm
Yes.
sorry. So, the allergen is a shape on the surface of the cell.
Yes.
And that you're not competent to that shape?
Right. You hypersensitive to that shape.
Okay.
So that's where you're allergic.
You're antigen, but you're incompetent.
You're hyper. So let's do the second question. So this one, different classes of antibodies. That's that big list you learned last time. But which one is the allergy antibody? IG who? IgE spells allergy. Right? IgEs are the allergic, the histaminic ones. So IgEs signify allergy. So number A3, something about mast cells. I don't have any pens. Let's learn a mast cell. Saladin goes on for a page about a mast cell. Let's simplify his life. A mast cell, in English, is a basophil. Tell me what a basophil releases?
Histamine.
In English, that means mast cells release histamine. The only real difference is a mast cell doesn't float around, a basophil does, but they all release histamine. So mast cells are histamine-generating cells. That's all that means in English. So what you're going to do now with your brilliant team is you're going to read through this scientific gobbledygook and then try to answer the questions that are the B's on the other side. We'll give you about 5 minutes to read that, and then we'll walk through the questions as a class. But see if you can read that gibberish, make sense of it. Future healthcare professionals that you are. I think it's fascinating, personally. Next term, we start breathing. We start eating, then we start peeing, then we start fornicating, and then we have babies and go home. That's what happens next term. See if we get stuff at the end. Sex and babies at the end.
Oh, it's mine. I'm a mess.
In the article, Biology 112, this is basically that. Sound spring. talk, you can do that.
Maybe someone died from a peanut allergy, but you would call the person that got the organ.
I love the understatement. You became unwell.
Yeah.
Cardiac arrest, coma, death. That tends to be a little on the unwell side. Yeah.
Yeah, and then they.
Laryngeal dyspnea.
And suddenly you don't without
Yes, this guy
I
died.
know. Just a little unwell.
You're feeling unwell. Yeah, that's pretty harsh saute sauce there. That's a Chinese food cart, right? It's very happy.
Yes, they don't want that.
All right, let's start going through these questions. Question number 1 asks you about an HLA, and this is nothing, I'm never going to put this on a final exam in your life, but HLA stands for human leukocyte antigen. Leukocyte means what? White blood cell. Antigen means what? Shape. So in English, the shapes on humans' white blood cells. That'd be $50, right? So, Biology 112 word, what does phenotype mean? What's displayed, or literally the shape in this case. So in English, HLA phenotype are the physical shapes of your antigens on your white blood cells. So if I were to draw, this is not anatomically correct, right? If I were to draw some, whatever, that's your HLA phenotype. That's the antigen on the surface of your cells. I'm just making up something there. So, the harder question then is this last one. How does an HLA relate to an MHC? Remind me what MHC was. Self. So in English, your HLA is your MHC. It's a version of your MHC, which is self. So in English, you can say self-antigens, you can say MHC markers, or you can say my HLA phenotype. They're all basically saying the same thing, the shape on the outsides of my cells. And why does the self matter? Why do you need to know your own shape?
You attack it.
So I don't attack it, right? I attack everybody else. So why would people with transplants need to worry about HLAs?
They don't attack it.
Very good. Let's go back to the gobbledygook. Notice that they kept talking about all these letters here. Anyone guess in chemistry what those letters are trying to describe? The shape of the antigen. So, if you wanted your organ to not be attacked, what would you need your HLA phenotype to be?
Match it as close as possible.
Exactly the same as the donor, right? Then your body would think it's you. But notice, are these people the same?
No.
No. So that means they're not the same looking cells. What does a T cell do to cells that don't look like it should? They kill and attack. So when they're looking for donors to match organs, they're looking at the HLA shapes to see if you match up. So who would be the only person on the planet who would definitely guaranteed to match you? Twin. Your identical twin. That's why you have them as for parts, right? So identical twin would have the same MHCs because you're genetically the same, right? So on a donor list, we're looking for how you match in terms of the shape on your MHCs. The closer you match, the more self we are, the less reaction there is. Make sense?
Do you know how much percentage it is for a sibling versus like
You and your parents would share no more than 50%. You and a sibling would share no more than 25% of your HLA, of genes. Now the genes could just by chance be similar, but you actually, a donor list would actually be probably closer than a sibling, just because there's more donors. A cousin would be, 12.5%. Some third cousin, eighth removed, would be basically 0. Yeah. Yes.
How close do they have to be for
Perfectly identical would be the best. As you move away from that, I don't know what the actual number is, but as you move away from that, there's less and less success. Make sense? So that HLA is just saying how close to you are, if that even comes out as English. So let's do B2 then. Tell me how an IG can be specific.
They're all specific.
They're all specific, right? That's the point, is they only have a shape that matches a peanut or whatever. That was your quiz question today, right? They all have different shapes. So the IgE for peanuts would be different than the IgE for strawberries. They have a different shape. Remember the specific immunity. Let's do B3 then. Ooh, we're looking for degranulation of basophils. What's happening when they degranulate? Releasing histamine! All right? That's what it is. Let's go back to that picture. For your lab exam tomorrow, tell me what type of blood cell a basophil is. Is it what? Granulocyte! Wow! That's what you see as the speckles on the slide are the granules of histamine. So they degranulate, they're releasing their speckles full of histamine. Or vomiting in English. Right? So they're degranulating when they release their histamine. Make sense? I guess. Good. Okay, so we figured out that it's IgEs, we figured out that you're getting histamine, blah, blah, blah. B4, why are they put on immunosuppressive therapy? Let's look at the thing here. That is, yes, that's right in this paragraph here. There's that cyclosporin from your quiz, right? Blah, 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 blah. So why do we have to turn off your immune system? They don't eat my organ. Why would they eat my organ?
It's not me.
Because it's not me. It's somebody else. And we're going to attack non-self. So you have to shut off the immune system so it doesn't attack you, or the not you part. Make sense? Yes.
Is that immunosuppressive therapy going to be basically permanent, or can it be gradually?
My understanding is yes, permanent until you die, which is permanent. So depending on how far apart these are, it could be permanent, or we can wean you off later, but it's going to be a long time. Yes.
Is there like a specificness to the immunosuppressants, or like?
There are. Cyclosporin shuts off everything. So you would have no B's, no B's, nothing. It's all off. This one I think is the same. The Maramimab, don't quote me, but I think that one does the T side. Don't quote me. Corticosteroids do kind of both sides, but to less extent than this one. So I have to go back to my drug index, but they're basically, you can target a B or a T, or you can shut the whole thing down. But that's basically your two choices.
Couldn't you teach your system to not to attack those? I mean the
Tolerate it?
Yeah.
You could if you knew how to do it. Problem is,
And since they learn.
yeah, the problem is you have to like inject the liver in your body over the course of years. Which I don't think do that. You're dying right now. Let's keep going. Number 5. Here's a big take-home message for final. Your initial exposure did nothing. The next time it got worse. Can someone explain to me why it's worse the second time?
Because it remembered it.
And what remembers it? What are we talking about? The memory cells. So every time I get exposed, I remember the last time. So I have more and more memory every time. That works also for allergies. Your first strawberry was fine, the second one was worse, the third one you're dead. Because of this concept, you're going to get worse and worse each time. Yes?
Is this the same with things that are hypersensitive? Like
Yes.
those will keep it-
Yep. So this graph here. So every time you get another response to the same antigen, bigger, higher, faster, stronger. So you're allergic to it now, you'll be more allergic to it next time.
Are you
So-
actually making more memory cells?
You do make more memory cells, plus the memory cells just get faster.
They just get better at what they do.
Yes.
So this is the same as RH antigens that was on the
Yes.
RH.
So first baby's fine, second baby not so fine, third baby doesn't make it. Okay. This is a vaccine, but the vaccine concept is the same as this. Right? Memory cells. Okay, blah, 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 blah. Let's go down here. So this is just gee whiz. So can anyone tell me in medical terminology C1? Pluripotent hematopoietic stem cell. Oh, come on, you did this. Okay, hematopoiesis. What's that mean in English? Making blood. So where do you make blood?
Bone marrow?
Bone marrow. Can someone tell me what pluripotent means? Makes a lot of different things. So in English, a bone marrow cell that makes a bunch of stuff. Right? That's what bone marrow does. So now what we're more concerned about is C2. What are those bone marrow cells doing when they undergo hematopoiesis?
It's recycling and making more?
Making more cells. So if you read through the whole article, they think they gave you basically bone marrow from the guy who was allergic into somebody. And that bone marrow remembers the allergy, and so it's cranking out the immune system of the other guy that was allergic.
Huh.
So you inherited not only a new liver, but congratulations, you're allergic to peanut butter. Kind of thing. But that's not the point of the article, right? So the point of the article is this idea of IgE, histamine, and the memory cells. They get worse and worse and worse each time. Make sense? Okay, so now I'm going to give you HIV. We're going to take a different angle on what we're learning next. So turn the page in your packet of pain. I'll give you some HIV. Yes.
Oh.
It's a weird, bizarre thought.
Oh, cool. The HIV cycle.
It looks so. I don't know if I have any. All my. Have a cow.
When farmers market downtown opens tomorrow.
Liver failure is failure.
And the I think I go to the
Liver failure tends to
every 2 weeks in the winter, but I don't know if they have the honey people in the winter. So they might not have in front of them. I don't want to go back to
Yeah, what they think happens is you heard
tomorrow and see if people are
somehow you remember, get remembered. That there's genetics for what you heard. Everyone have their HIV? Hey, so I'm not going to ask you on the final to like go through step by step by step, but we're going to walk through it and learn it step by step, so you can understand the concept of HIV. So let's start at number 1. There's the virus floating in your body, doing bad things. Look at number 2 and tell me in English what that means. Who binds the what?
The virus binds to your helper T cell.
Very good. So the first thing you have to figure out is what this gray thing is. That's a helper T cell. So that gray is a helper T cell. Tell me again what helper T cells do. They command, they do all that, right? So the virus is stuck to, binds to, recognizes, whatever, your helper T cells. So hence binding and entry. Okay? So what do you see happening in step number 3 after the helper T cell and the virus have sort of connected? Stuck together and sticks its stuff in, right? So the virus is injecting its genetics into the helper T cell in step 3. Now step number 4, the group of Truvada just mentioned this to you. How nice. Can you read step number 4 and tell me in English what the virus is doing? What part of it is duplicated? What's it doing? Very good. If you read through that, it's taking its RNA and making DNA from it. Biology 112, you learn the central dogma, right? Central dogma says you start with DNA, you make what? RNA, and RNA makes what? So this is in reverse, right? They're going RNA to DNA. So hence we call it reverse transcription or retrovirus, because you're going backwards the way we think of biology. Right? So HIV is a retrovirus because it does step 4. You have reverse transcription. So in English, it takes RNA and makes DNA out of it. That's what step 4 in English is trying to tell you. So look at step 5. What happens after making DNA in step 4? Integration. The board collection. Right? You're now 1. So virus information is now inside my information. This is my nucleus here. You're now 1. So look at 6. What's happening in step 6? Who's making one? Who's making that? My cell is making that, but what is that thing I'm making? What is that? That's virus, right? So my cell is now making virus for the virus, right? The virus put its instructions in, and I read that and make viral particles. So then if we take 8, 9, and 10 sort of together, the baby viruses come out. Aren't they cute? They grow up, and now number 10, they're ready to go. What are they going to do after step 10? Go back to step 1 and do it again. So we find a helper T cell, we squirt it in our guts, we make a DNA, we merge with them, we tell them to make babies, babies come out, and we repeat. Lather, rinse, repeat, right? That's the basic HIV retrovirus pathway. Okay? So if you understood the basic pathway there, find your sheet on HIV. We're going to kind of walk through this as a class. So tell me, number 1, how does HIV know that's a helper T cell? Gee, those are things on the surface of a cell. What do we call things on the surface of a cell? Antigen. Okay, so the antigens or receptors or protein shapes, or pick a word, but I like antigen just to beat it into it. So it's looking for specific antigens on the surface. That A word again, right? So tell me, how does that relate to a protein?
They are proteins.
They are protein. Shape, remember that from Biology 112. Things on the outside of a cell are protein. What's that to do with genes? Genes make proteins, hallelujah. Okay. So, your DNA, your genes, make your proteins that give you the antigens. So you genetically look like this, and the virus genetically looks like that. Make sense? It's all about your genes. What's number 2? What cell is it that it's infecting? The helper T cell, also called the CD4 cell. The helper T cell is a good plan. So what's the job of a helper T cell?
To turn on the rest of the immune system.
Turn on the rest of the immune system, give orders, regulate. There's lots of words you can throw at it. Let me show you this picture here. I lost them now. Where'd it go? There it is. So just another way to look at it. Remember, this is our helper T cell that reads the antigens, gives orders, tells on T's and B's to clone and do their job. So giving orders, giving instruction, command, whatever. Make sense? All right, so now, what happens to the helper T cells when the new HIVs are coming out in step 8? Read step 8 and predict for me what the helper T cell would do when step 8 occurs.
It kills it.
It kills it. Very good. If you read step 8, the virus comes out, excuse my voice, takes the cell membrane with it. What happens to cells when you take off their membranes? They lyse, they die. That's the whole complement idea. So your helper T cells die off when the HIV punches holes in it as it leaves. Make sense? That's seen in aliens, right? That's the concept here. Something comes out of your tummy, you die. Let's do question number 3. It's a retrovirus. What step on this chart tells me it's a retrovirus? Step number 4, that reversal step. So if you do step number 4, you are a retrovirus. You go RNA to DNA. You go in reverse of our central dogma. So tell me about other pathogens, like bacteria. Do they do reverse? No. What about worms? No. So, and not all viruses do, but retroviruses do, yes.
So what are prions?
Good question. Prions are proteins that act like a virus. But they're not even they're a single chemical. Yes, freak out time now. Something that's not even considered a cell, or even a living, or not even non-living thing. It's like a rock that kills you. Literally. Okay. So don't get this idea of reversal. So it's just that step 4. All right, so question number 4. How long does it take to go from integration, step 5, to budding out, step 8? 3 steps. That's what someone said this morning. That's not what I'm asking. Time-wise. How long is that time-wise between number 5 and number 8?
Does that vary from person to person?
It varies, that's right. We don't have a definitive answer. It varies by people. So for some people, that could be weeks, months. For some people, like Magic Johnson, claims it's decades since between 5 and 8. The average is roughly 6 months, if you average it out. You know that from when your HIV tests are due. How often do you have to go in for an HIV test? Every how long? Approximately 6 months. That's the logic behind it is after 6 months we should see 5 to go to 8. But there is no definitive answer. It varies.
In that 6 months between tests, when someone keeps waiting to manifest, are you still able to transmit it or not?
Yes. If it's free, if some of it is budded out, which may or may not have occurred, then you could transmit. If none of it's budded out, then in theory you wouldn't. But since we can't test you very effectively, we don't know. Make sense? So that's a variability, right? We don't know. But number 5, HIV and AIDS. So tell me when someone's HIV positive, like on the case study tonight, what's that mean in terms of our chart? What step do they have?
It means that it's in the cells, but it hasn't put it out yet.
Say number 1. If you have step number 1, you are HIV positive. Virus has entered your body, you are HIV positive. You have HIV. So 1 on, basically. So to get to AIDS, what steps do you have to do to get to AIDS? Yes, basically 8, 9, and 10. Baby viruses have come out and are now killing off your T cells. So you can be HIV positive but not yet have AIDS. You have virus in you, but it's not destroying things. Everyone, well, almost everyone with AIDS is was HIV positive, yes.
So, sorry if this is a stupid question, then why do HIV positive people why are they immunocompromised if it's not in their T cells yet?
Let's find that out. That's the next question. Let's look at number 6. Why do people with AIDS have these wacko infections that no one else gets?
Because their T cells aren't functioning.
Right, let's go back to our T cells. If I kill off my T cells, helper T cells, what do these other cells do? Absolutely nothing, right? Because I'm not activated. So hence I get infections, I can't remember them, learn them, or attack them. So hence you die of whatever.
But if you're just HIV positive, if you don't have AIDS yet, then why are you still immunocompromised?
You're not formally.
Okay.
If you're just positive and it hasn't lowered your T cell count, then you're just pre-AIDS, they would call it. You haven't lost your immune system yet. Then there's ARC, which is your immune system's beginning to fail, then AIDS is actually failed. So formally, the way I learned it, you can be HIV positive and still have a fully great immune system. So only when HIV kicks in, begins to do step 8, 9, and 10, then your immune system is now compromised. So formally, yes.
So when you accidentally get pricked, like at the hospital,
Yep.
with someone who has HIV, they give you medication.
Yep.
So what is that supposed to do?
We'll do that next. That's a question. Come on.
Oh, I'm sorry.
Okay, yeah.
Have they tried formulating like AIDS drug that like just activates the immune system? Like just is like, okay, helper T cells are gone, but we're gonna do this other immune system activator thing?
They've tried different things and none of them seem to work outside of mice.
Mice.
Yeah, so interleukins was a big to do early 2000s. Interferons was big in the 90s. There's other drugs. They're all going to be magic. And then they realize you get to people and there's no magic. So, yeah. Yeah.
So when it is 8, 9, 10, then you have AIDS.
When 8, 9, 10 has occurred, you're now going to begin getting AIDS. Depends on how many of them bud out.
So when is like how do they know when like you have
Oh, glad you asked, because we have that question coming up. Look at number 7. How do they know how bad you have HIV or AIDS? What are they going to look for?
T cells.
T cells. Which T cells are they going to look for? Helper T cells. So they're going to look at a graph kind of like this. They're going to have your CD4 count or your helper T cell count or whatever. So to get your nomenclature right, these CD4s, those are helper T cells. These CD8s are cytotoxic T cells. So look at your CD4 count. What's happening to your helper T cells when HIV is going gung-ho? Right? It kills them, right? 8, 9, and 10. So as HIV is killing off my helper T cells, the number should just crash like a rock, right? Look at my cytotoxic T cells. What did they do? Nothing. So a very common misconception people think is that when you have AIDS, you don't have an immune system. You have a fully happy army. Your army is full of ranks. What you don't have is anyone to command them. So they're just sitting there cleaning their gun. Right? There are no orders, they don't know what to do. Right? So in number 7, they diagnose you by looking at the CD4 count and the helper T cell count. Yes?
Could a blood transfusion help elevate your CD4 T cell levels?
Yes. They did, I don't remember what year it was. It was in Germany, believe it or not. They did an experiment where they basically drained a guy who had HIV. They just, like changing oil in a car, drain him, and give him a totally new batch of blood from a donor. And they found out that he didn't have any active HIV afterwards. The
Wow.
problem with that, if you think through it, is how do you drain somebody like effectively? And then that doesn't mean he doesn't have HIV in the blood. They don't get out every bit of blood, right? There's no way to get every drop out. So you're reducing most of the blood, but there could still be HIV in the other blood, right? And that's the catch is, would this work in a population-wide? Probably not. But it did seem to work in kind of in theory. But there's no way to verify then if that would work forever.
Was this guy voluntary or?
I didn't read the article enough to know this. I just read the results. I don't know. I assume he was.
Is he still HIV?
To my knowledge, at the time he was. I don't remember if they did a follow-up 20 years later. So let's do this one. Number 8. HIV cannot fix mistakes. Meaning, when it reproduces, what will happen to its genetics if it cannot fix a mutation? They'll be different, right? They'll mutate, they'll change their DNA. So, let's talk about vaccines. Would that be, could you make a vaccine if the enemy is different every time?
No,
No,
essentially.
because vaccines require you to remember what you saw. I can only remember the one before it changed. So, the idea of a vaccine is not very realistic for most of the time. At least the common way we do it. Yes?
So basically what that's saying is that if it differentiates really rapidly without
Yes.
affecting its impact on your body,
Correct.
that's what makes it hard to do a vaccine.
Right. This and the flu are basically the same kind of idea where you're chasing after it because it's changing faster than your body can keep up. It's the same logic. To show you I'm not making this up much. Let me show you a picture of the different families of HIV. Not that you ever have to know this, just you as trivia. Show you a picture here. Yes. So the group mentioned it with Truvada a little bit. There's different families. You never have to know this,
Oh
right?
my goodness.
But HIV, there's 2 major groups, species, HIV 1 and 2. 1, which is the older one, breaks down into groups, and then those break down into subgroups, also known as clades. So you could have a HIV 1MA in your body. Your neighbor could have, MC. They're not the same HIV. They're distinctly different in terms of their shapes. So your vaccine would have to include every clade with every group with every species if you want to cover everybody. Right? The irony is, this is worthless trivia, different countries have different proportions, different cities have different proportions, and people can be infected with more than one species at a time. So a vaccine for Seattle would not work in Portland. They're different clades. Right? Assuming that you even got the right one anyway. Right? So that's why measles you can vaccinate for, there's only one measles. It's easy. This is harder because you have multiple types that are mutating.
And if you get a vaccine, won't you run the risk of contracting the virus?
You can make it a deadened virus, like a flu shot. But then the other thing is, what happens if I give you the vaccine, there's a new one that comes up, and that's the problem. So again, this idea of the vaccine, at least the way we think of it now. Fine, turn the page. Let's walk through the diagram and figure out where you could stop, control, alter this HIV. Let's start with step number 1. Can you control step number 1? Say yes.
Yes.
Don't catch it, right? Exposure. If I don't have HIV in my body, I don't have step 1, and then we don't have to worry about it. Imagine that, if only everyone knew that. Let's look at step number 2. Can you do something about step number 2? Yeah, not by choice per se, no. Although, to be honest, there are people whose antigens are a different shape than the HIV combined with. So what would happen if you genetically have different antigens than the HIV combined with? What would the HIV do?
Be immune.
You'd be immune. The HIV could never find your cells. So there are people on the planet who genetically are immune to HIV. HIV cannot go to step 2 if it can't find you. Yes? Usually genetic immunity or natural immunity of HIV is what they call it. Yeah.
But they still have HIV, so they can give it to someone else.
That's exactly right. So they're carriers, they have HIV in their body, they're giving it to everybody else. Yes?
So are some people more susceptible then?
Yes, that's the logic. Is some genetics make you more susceptible, some genetics make you less susceptible. Problem is, unless I expose you, I don't know which one you have. So that's the problem with it, is they find the people post facto. They happen to all be prostitutes, in case you're curious.
Oh my gosh.
But what happens with these prostitutes is they should all die of AIDS. They never catch anything, so scientists spend money to go investigate their genes. right? So you should be dead, you're not dead, that means you're immune.
So what region are people protected?
The prostitutes they found were in Zaire, Tanzania, I think it's either Thailand or Cambodia, I can't remember which one it was. There's a small subgroup in Australia, and there's one in Kenya. The ones that I actually went on.
Is it
Don't
from?
go there planning a vacation, right?
Is it because of the mosquitoes? Is that why?
No, the Australians wouldn't have that. So these are people that should be dead, they just found them out. So all of you could be immune to HIV, but unless we expose you and test you, there's no way to verify. Right? Well, let's assume that that's not your cup of tea. Can anyone see another step on here where maybe you could stop something, like the presentation tonight? 4. Step number 4. You read step 4, you see the magic E word, which is enzyme. From Biology 112, can you stop enzymes?
Yeah.
Sure you can, right? It's easy to do. So if I stop step 4, the virus cannot integrate and keep going. You still have it, it's still docked, you just can't go further. Truvada was that. It was supposed to stop it from doing step 4. Can anyone see another enzyme somewhere on this page?
5.
Step 5. There's an enzyme required to integrate. So if I can stop that enzyme, which Merck says they can, then it wouldn't integrate into me. Keep reading. What about step 6? Do you can stop step 6? Say no. How about 7? Say no. How about 8, 9, and 10? Say yes. What do you see in 8, 9, and 10? Protease enzyme. So if you stop 8, 9, and 10 by stopping the protease, the virus wouldn't grow up. Peter Pan.
So basically, enzymes you can change. But
Yes. So, if you know somebody's taking HIV medication, they call it a big dose of cocktails, right? Can you guess why they call it a cocktail? It's a mixture of all these enzyme drugs that you're trying to stop. You're trying to stop step 4, step 8, step 10, so you're taking a cocktail of different medications to stop different enzymes. Right? Stop it from going in, stop it from coming in. That's the logic behind HIV. Currently, I'm aware. Make sense? Go out the enzyme. So let's go do these little bit of, Okay, so where do you think HIV is the highest rate? Did anyone take a guess for me? I vote for Sub-Saharan Africa. Anyone want to contradict that? Let me get the official map up here for you. Where's it at? There we go. Bring it on the screen. Yeah, it's going to be small. I just, for some reason, can't get this image to get bigger. No, you don't have to draw this, right? But the way this works is the darker the color, the more HIV you have. The dark, dark, dark is roughly 30% or more of your population. Adults, women, not children, but adults, men and women. So where do you see the darker colors? Let's see. Out here now, right? Sub-Saharan Africa. Where else do you see some dark colors? Yeah, Putin's got another problem. Here it is, this is one. Southeast Asia. What Suriname? Is that what that is?
Congo does not report.
That's the trick. These are reported to the UN. So they're self-reported. So notice how China claims they have less than everybody. India has none. Maybe they do, I don't know. But these countries report the data to the UN self-reporting. So you could lie through your teeth, you could be honest, you could make it up, you should just not care. Right? So don't plan your honeymoon based on this. But, so we're sort of lower end, middle of the road kind of a group. Right? So let's figure out why. Why would Africa have it worse than everybody else?
Lack of resources and education.
Sure. So there's lack of health care and education, the internet, you know, if you have sex enough time to be cured.
Lack of government.
Lack of government, so if you're in a civil war, who cares about HIV? That's right. Cultural practices, dry sex, no sex, lots of sex.
Didn't it originate there?
Requires blood transfusions, bunch of stuff.
Didn't it originate there too?
Yes, I know of an article that said the earliest known case they found was in 1955 in the sailor that sailed out of Senegal. So it's been around since the 50s.
Oh, wow.
It's probably earlier than that. So, but the gist is that it's mostly Africa, Southeast Asia. So how do these people get it? How do all these people in Africa get this thing? How do they transmit it?
Generally sexually transmitted.
Usually sexual disease. Right. Yep. So sexual transmission. They think that's probably the Southeast Asians, the sex trade. They think this one actually is probably IV drugs, maybe. I have no idea why these people have it, right? But they think it's mostly sexual transmission. So now what we're going to do is go through our risk factors for these activities. So are these bad for you or good for you?
Oh my
Let's start with number
goodness.
1, transfusion of a contaminated blood product. What kind of risk is that? It's high, right? Because I'm putting HIV into you. That's step 1, guaranteed by volume, right? Okay, needle sticks.
High.
Medium.
The CDC says actually it's low. They do. It probably was higher in years past. But what do you do differently now than you did 10 years ago at your work? Don't share needles, right? Wear your gloves. Safe needles, you got a sharps container. The other thing they found out depends, well, how much of the HIV was on the needle that poked you? How deep did the needle go into your body, right? All those are factors. The CDC says you're not as high at risk as you thought you were, but you're still at risk, right? EMTs especially, right? Mosquitoes. Low. They carry HIV, they do not transmit it, believe it or not. So there's a lot of money spent on mosquito spit. Yeah, but low. Breastfeeding.
Low.
The answer is we don't know. There's no official number. Because, to do that, you'd have to intentionally infect milk and have the child eat the milk, right? That's kind of unethical, right? So breast milk does contain HIV. So if you're drinking breast milk, you will have HIV, step number 1. The trick is that it actually get into the baby's bloodstream to find the helper T cells, right? And how much did it get in there? So we're not sure. How about perinatal exposure, placental during birth? We still don't know, actually. Same reason is, well, does that mean it crosses the placenta? Did you get it from mom? Did you get it from someone else? Right, so they don't know.
They just schedule C-sections.
So in the US, we do C-sections. The idea is if I lift baby out, there's less exposure than if baby's coming out of the vagina. Things tear, things rip, things bleed. There's more exposure. Vaginal birth. Now, if you're in Africa, that's not a choice, right? But that's the idea. How about kissing?
No.
Hopefully it's low, right? So there is HIV in saliva. It takes gallons of it to infect you. You have to basically slobber on somebody. You have to have an open sore all over your mouth. Why the hell are you kissing people like that? Right? How about French kissing? Again, low unless you have open bloody sores on your tongue.
Gross.
Casual contact, again, usually not. Toothbrushes?
No.
Low, again, unless they're dripping with ooze.
Ew.
How about dried blood?
No.
Low. HIV does not like air, so it tends to die when something dries up. So it has to be liquid body type stuff. Okay, let's have some sex. Okay. Anal sex with infected males.
High.
It's considered a high risk. Body fluid, direct contact, the anus has a lot of blood in it, so therefore immediate contact. How about vaginal sex with infected males?
High.
High as well. I think the female reproductive tract has blood in it. Semen goes in. If you're on your period, that adds a little bit level of blood contact. So it's easy for the HIV to find its way to number 1. How about vaginal sex with infected females?
It happens.
It does happen, but it's medium. It's less risky than the other way. Why would that be? Why would a woman have a harder time giving to a man than a man to a woman?
Because the man injects his hand.
That's right. So if you think about sex for a second, the man puts his fluid into her, not the other way around, right? So it's harder to get the HIV in contact with the man's helper T cells than the other way around, right? So there is that thing where it's a reduced risk for male and female. How about oral sex on a man? It's considered low, believe it or not. And oral sex on a female?
No.
It's also low. Takes more fluid, harder for the virus to find its way. Let's do the last one, which is so fun. Let's say you have something else like syphilis, gonorrhea, chlamydia. What's your risk? High. In fact, it's higher than the others. 8 times higher than any other risk group. So why would that be? So your immune system's fighting something else. What else?
It shows you pretty risky lifestyle.
Risky, stupid behaviors, right? And give me the symptoms of most STDs. Oozing, slimy stuff in your genitalia. Not that I would know, I have a friend, right?
That males who are circumcised actually think the CDC said that they're like up to 60 or 70% less likely
Yes.
to contract
In fact, I think wasn't it Zaire that now mandated circumcision for women? Because it does reduce transmission by almost half.
So,
Because there's less place for the virus to live in the skin, under the skin.
I know the Maasai in Kenya don't circumcise until they're 13.
Oh, that'd be fun.
It's pretty barbaric.
Yeah.
They actually got so, I guess, powerful. I mean, the Foo Fighters were involved, but they went to South Africa and they spoke with the president after Mandela, and they got him to become an HIV denier, and it ended up killing, and infecting
Yeah, that makes sense.
hundreds of thousands of people.
There's a lot of that. The internet's been fabulous for people that have wacko ideas. There was something, not that I'm defending this, there is something where they took some data and they ran with it the wrong way. If you take, where's this trivia? If you take cocaine, HIV kills you quicker. HIV amplifies, cocaine amplifies HIV reproduction, so step number 6. It also amplifies how many come out. So cocaine addicts die of AIDS quicker than everybody else. That's also why the San Francisco group got it so fast, right? Gay men were shooting cocaine in San Francisco. So what people did is they thought the cocaine must be what does it, not the HIV. So they started thinking it's drug abuse, not HIV. Well, no, the HIV, the drug use amplifies HIV. It's a lot of them.
So meth is
Meth makes it
kind
worse.
of big in
Yep.
vaccine.
Anything that's a stimulant will cause HIV to increase speed. That's all I know. Yeah.
So, just having, what makes you more or less infectious? Basically, if you're at still at kind of before step 8, 9, 10, you're less infectious to others?
Depending on how much HIV you have in you, yeah. So once the virus buds, now the virus is amplifying. That makes you more. Make sense on HIV? Okay, so we got one more little thing to do to wrap this up here, which is this one. We're gonna learn a little bit about flu, because HIV probably won't kill you, the flu will. Alright? So we're not gonna do all the questions, we're gonna zip along here and do a few of them. So we're gonna start on, number 1 from Biology 112. Number 2, what is an antigen? You think that's important? What is an antigen? Shade marker. You bet your bottom dollar that's on your final. That's a very important concept. Is that antigens are shapes on cells. Now, if you take micro, you're going to learn number 3 a lot more. But there are antigens specific for flus. You've actually heard it in the news. Can anyone name the 2 letters of antigens on a flu? H and N. So a virus, a flu, has H antigens, hemagglutinin, and N antigens, neuraminide. So if you read the news and someone's talking about, what, H1N1, what they're talking about are the shapes on the flu. So someone give me another bunch of shapes. H what? H5N1, H2N3, right? These are all different antigens on the flu. So you can think of them as different shapes, different markers, different species. So the H and the N are the shapes on the flu, the antigen.
Does the number correspond to how many are?
No, it's actually just the category class. That's like how they found them. Right? Make sense? So let's go down to number 4 then. Can anyone guess from Biology 112 what antigenic shift means in English? Mutation or change in antigen. So flus can shift their antigens, meaning they change the numbers after their H's and their N's. So let's say you're vaccinated for H1N1 and it mutates to H1N3. What just happened? You've lost your memory, right? And therefore, guess what? You got to restart the whole ball again. So one of the problems with flus is they keep changing their H's and N's. Therefore, you lose your memory and have to start over every round. They do this very, very quickly, very, very well. Make sense? Okay, so let's go down through this part. So you read this and you get flu shots, blah, blah. What's in a flu shot?
Antigens?
Antigens. How many usually?
2.
Usually 3, except the one that last year, which was a 4. So usually they take 3 sets of these letters and stick them in your shot. Welcome to Vegas, right? They're hoping the virus you find is the same letters as these. So let's figure out our friend. My friend got the shot and got the flu. Explain that. How could you get the shot and then get the flu right afterwards?
Because it has a different kind
Could be a
of.
different strain, so you weren't
Yeah.
vaccinated for it, or you didn't have time, Let's go back to that magic picture.
Or your age, like lower.
Could be lower than where's that picture at? My memory's off. There they are, right here. Right, there it is. So remember, it takes about 2 weeks or so. So you could have, what we all do, you wait till you're sick and then visit the doctor, right? I went and got my flu shot as I'm coughing up the flu. That doesn't work, right? So you could have already been sick. You could have been a different strain. You could be immunocompromised or old or a baby, whatever. Yes?
Could your body just be overreacting to the dead cells?
It could just be something else, like an allergy, or it could be a stomach flu, which isn't a flu, right? Stomach flu is something different. So when people say they got a flu shot and got sick, chances are they got a flu shot, but it wasn't what made them sick. 9 times out of 10. Okay, yes, go ahead. Oh. Let's do number 3 then. The joy of the flu. Other things can get it. Name something that gets the flu besides humans. Birds get the flu. Avian flu.
Pigs.
Piggies get the flu. So birds and pigs get flu just like you.
Is there a reason why?
The quick answer is receptors.
Okay.
The longer answer is magic. But so let's go through this. So we're going to go back and do some avian flu, right? They killed all the chickens in Hong Kong. So what we're worried about is basically number 2. Why did the avian flu, or the flu of 1918, which killed 22 million people in 6 months, differ from yearly flu? What was so unique about those bad flus?
Because it.
Very good. It killed people that were in good health and middle age. So who usually dies from a flu?
And
Really
older
young,
people.
Really old or immunocompromised. Turns out that those flus kill off 18 to 40 years old prime health. The soldiers died first. Right? Why? And that goes up to number 3. Tell me from your notes what cytokines did.
So the ones that reproduce, or they send out the signal to reproduce, like the proliferation of B cells and the T cells.
Very good. So cytokines activate the immune system. Right? So they activate your B's and T's. So if you're very healthy, how many cytokines can you make? A lot or a little? A lot. So what would happen if you made too many cytokines because you were such great shape and this number went way, way up? What happened to your activation? It could go way, way up. And what do B's and T cells do to your body to kill the invader? What do T cells do? They kill your cells. So if the invader's in my lungs, I have too many T cells anxious to fight. What happened to my lung cells? They start attacking my cells. And what happens to my lungs when they're losing all their cells? So they stop. What happens to you when your lungs stop?
You die.
You're dead. So what's irony here is that your immune system is actually too good. You ended up with such a good response that you kill off your own body while you're trying to destroy the virus. That's called the cytokine storm.
Why did they just choose to ignore the cells? We don't.
There's a virus in there and you're going to kill it. So the problem was people that were in prime of life had an overactive immune system, a good immune system, so they end up with cytokine storm. Grandma and grandpa don't have a very good immune system. They actually live through it because their immune system never turns on enough to kill them. So the problem with the swine flu is it caused you to do that. You overreact to the flu. Make sense? Yes?
So does that mean they get treated to some extent with like immune suppressors?
If you live long enough, you could do that. The problem is usually they died in about a week, but that's it. Make sense? Yes?
So the flu makes it like that's the reaction you're having? Or is it
Yep.
the cause?
And to be honest, your flu doesn't cause any symptoms. The immune response is what causes all your symptoms of flu. The malaise, soreness, the flu is the cough. That's the immune system destroying your body to get the flu out. So the better your immune system, the more symptoms you end up with. Same
That's
backwards,
promising. After you have it for 4 weeks.
Yes. So, yeah,
So
in this case. Because the symptoms are a result of the immune system attacking. All right, so let's figure out number 4. Yes, okay.
Is that same kind of reaction to regular seasonal flu? Or is it
Seasonal flu causes less of a storm. So you still react, you feel lousy, but it tends not to be overwhelming like the cytokine storm.
but the same general thing, like the better your immune system, the worse it's going to be.
Basically, yeah.
But it should be over fast.
Should be over faster. Right. So, let's do this one. How could you end up with flus all these different years? Different strains. They've mutated, and so you don't have memory anymore. You got to restart. Right? That's why they call them all those names. So let's look at number 5. To scare you, swine flu's H1N1 and avian flu's H5N1. So in English, it's saying what would happen if the swine flu and the avian flu had babies? What
Wow.
would the babies be? Give me some numbers. Why not? Right? You don't know. This is Vegas. You have no idea what those numbers will be if the flus start to breed. So hence the problem is, if you end up with these animals next to each other and they reassort, you make new flus every round that you have no memory for, which leads us to number 6. All right, here's where you should get nervous now. All right, so humans have a receptor on their surface called 2,6, and then we're going to ask you in the final. Birds have a 2,3, piggies have both. So tell me why you shouldn't go to Asia during flu season.
Because it's full of pigs and birds.
And people, right? Yep, so you have people, a billion people living with their animals in their house, which happen to be pigs and ducks.
In their house?
Yes, or close proximity.
What they worry about is that the avian flu will go into the pig and it will reassort in the pig.
Yes.
And then that's when it's going to become a human contagious.
That's right.
Is that because they have a similar receptor?
Yes, so piggies can be infected with birds or humans. Then they would do this, they'd reassort, then it'd come back out to humans. You'd have a half bird, half human flu that's now going to affect people, which your immune system has never seen ever in its life. So now you're dead.
So what does reassorting actually mean?
It means you take the genetics and you combine them different ways. Make sense? But this is again that problem with memories. You never see the flu before, so you're going to have a problem fighting it off. Everyone nervous now? Yes. Yeah, that's the idea of flu. So.
But hasn't that been going on for a really long time?
Yes. The problem is, as people get closer together and it's easier to travel, it becomes more of a risk. Now, it used to be you die of the flu before you got out of your village, right? Now you can hop on a jet and be in New York in 5 hours. Spreading it to everyone on the plane, and then spread it to everyone in the airport, right? That's the logic. Is it's just you move faster than the flu can kill you. Not that I'm nervous at all. I have found kind of property. Right. You okay on this? So again, this is a vaccine memory type of idea. As you change the pathogen, you have to relearn the immunity every time.
How do drugs like Theraflu work to slow down?
They don't.
They don't?
They don't do anything about the immune system per se. They help treat the symptoms. Like an antihistamine, you remove the phlegming, the expectorant, get out the cough. But they don't do anything really with the immune response. They just make you feel better while the immune response is going on. It's like chicken soup kind of stuff. Make sense? Questions on this? Let's talk about your final exam since you're sitting here so nicely. I wonder what will be on it. Good thing. Let me go through your final. All right, are we ready? Okay, the first page is mostly brain damage questions. So, if I damage my frontal lobe on the left side, what would I see as a person? I can't see what part of my brain, so those symptoms, the body part, the symptoms. Okay. Grandpa has a stroke. Classic question. All right. There's one question on the integrative brain areas, meaning basal ganglia, the RAS, not the RAS. Yeah, the other RAS. Right? The 1A RAS. Yeah, very good. Those things. So Jonathan rips out his basal ganglia. What do you see? Okay. There are 2 questions on the cranial nerves. Thou shalt know name, number, type, and what it's for. Those damn things are back. They never go away. Right. I have it. Yes.
Can you put irrigation on that?
Yes. So where it goes.
Okay.
There's a CSF flow question on there. Know the pathway of CSF. Where it's made, where it goes, and what I can do to mess that up in your life. There are about 3 questions on the parasympathetic and sympathetic systems. So everything about those. The wiring, the neurotransmitters, the effects, alphas and betas. If I block, turn it on, what would you see? All that fun stuff. All right, I have one question on how you see, or more specifically, how it can mess up your vision. There's one on how you hear, and there's one on how you balance. Jonathan ripped out his eardrum, kind of thing. Okay, then we have a whole bunch of questions on endocrine health.
Endocrine.
Although there's no chart this time, it's just questions. So I'm going to make it harder for you. About a page. Okay. So this includes, target, source, all that type. There's also one about hyper and hypo. And also include that is the chain of command, the releasing, the tropic, and all that. So this one's high. What's this one going to do? By the way, there's also a graph on it, just in case you want to know. Graphs. Then we have some blood questions. There's one on how you make blood cells. You can't write EPO, right? There's one on how you get rid of blood cells. And there's one on the transfusion of blood cells, how you type them. What can you give to whom? There's one question on the clotting factors, thou shalt know in order, thou shalt, in Roman numerals, thou shalt. Then there's kind of half a page-ish of heart. Thou shalt know the pathway through the heart. Thou shalt. Bless you. So what chamber are you in? What color is the blood? What valve is there? What valve is that? Is it up or down, dub or lub, or whatever? And that also includes the EKG. What happens during QRS? What happens during P? So B blood, UR blood, go. All right, there's a cardiac output question. Just one at this point. And there's a blood pressure regulation question. That kind of ends the review stuff. Now we're going to do new stuff. So we have a barrier question on the body barriers. There's a couple questions on lymph, the nodes, the flow, what it does, why you have it, what happens in there. There's an inflammation question about what makes you inflamed. If you haven't remembered histamine by now, do so. And a whole page and a half of B cells and T cells. So people will walk me through step by step how you turn them on, what they do, how they know to do that, the vaccine, the memory, and the IGs that go with them. So reproduce a couple pages of notes for me. To my knowledge, that's all I have at this point, which I'll finalize it over the weekend. So, nothing directly on the HIV chart, per se. I'm not going to ask you like step 5 or anything. If you understand the HIV, that would be something you need to. That's going to be around 80. So I have to tweak it a little bit. 75 to 80. Make sense? So next time you show up will be your final, and your grades are in the mail. Question? Concern? Hang-ups? Roasties? Okay.
The complements are activated by
I learned from the antibody binding because the plasma cells are your flexible initiate, but it actually the antibody is not